Okay, well, thank you all for coming. I want to welcome you. My name is Brian Somm. I chair the Department of Physics and Astronomy here at WSU, and I especially want to extend a warm welcome to all those who from other departments at the university and from outside the department that we don't normally see. It's a very great occasion for us to welcome again an honored speaker, a Nobel laureate. And I'll get to that in a moment. I'll let Professor Kuzik introduce our speaker. I just want to say, again, I want to say a couple words about S-Town Stephenson. This is the S-Town Stephenson Distinguished Lecture. It's also part of our Distinguished Colloquium series, the last of 5 that we've had this academic year. And this honors the late S-Town Stephenson, who came to WSU from Yale University in 1934. He did research in the physical sciences, primarily noted for his research on radar. He was also a very accomplished administrator at the university. He served as chair of the Physical Sciences Division, dean of faculty, and vice president. And I like to point out that he was also the founder of the honors program at Washington State University. And so we've had a series over the years of very well-known speakers that include Stephen Chu, Eric Cornell, Eric Mazer a couple of years ago, and so this continues in a tradition of bringing some very great speakers here to campus to deliver a talk. And so with that, I do want to mention that this is a distinguished colloquium. And one of the things that means is that there's beer and goodies afterwards. So I just want to point out that we have a reception at these. The point is that you have an opportunity to interact with and meet the speaker and to socialize yourselves. That was part of the whole point of this series. So I hope you will partake and enjoy that. And with that, I'll turn it over to Professor Kuzik for the introduction.
Okay. Well, it's my great pleasure to introduce our distinguished speaker today, W. Moerner from Stanford University. He started out born in California, but for some reason he went to St. Louis for his undergraduate studies at Washington University, where he got a BS in physics, a BS in engineering, and a BA in mathematics. So I'm doing this off the cuff, so hopefully I'm getting that right. And then he went to Cornell and got a master's degree and PhD degree in physics. And then after that, he went to IBM, and that's when I first met him, way long ago when we were young and I had hair. He's always had hair, but it wasn't real back then. And so he did work at IBM on single molecule work, which has transformed into what he's going to be talking about today. But he was also working photorefractive polymers, which was our area of interaction. And we actually have a common child, Dina Poga, who was my first graduate student who went to work in his lab at IBM. And then after IBM, when he was thinking about leaving, I have to tell this secret, that he sent his resume saying, hey, I'm going to be going to academics, do you have any openings? WSU was having a hiring freeze, so we couldn't consider that possibility. But he went to San Diego, University of California, San Diego, and then Stanford, where he's residing now. Now, one of the things that I want to point out to the students is that you can see that his training is in physics, mathematics, engineering, but he does multidisciplinary work, and he's in the chemistry department now, and there are applications in chemistry and biology. So when you get a degree in physics, you don't have to stay in physics. You can kind of meander around and get into other areas. But one other thing, which I remember hearing somewhere, I don't know if it's true, but I don't want to embarrass him, but apparently he has been a straight A student since kindergarten. Is that right? Yeah. Oh, yeah. So he's been at the top of his class, the top of his profession, all of his life. So it's a great pleasure to have him here. We're happy that he came to honor us with his presentation. So with that, it's yours. And I'm going to go to the back because I can't see well if I'm up front. So it's not like I'm trying to get front.
Okay, well, thank you very much for that wonderful introduction. I'm particularly indebted to Professor Mark Kuzik for inviting me here. And it's been a fun day, a great pleasure to visit WSU and a number of departments here. And so I'm going to tell you about our favorite area, single molecules and super resolution. I want to begin with a quick roadmap, though, and noticing that we're already starting late, so I'm afraid I'm going to run over a little bit. But in any case, I'll do my best to not run over too much. So I'll talk a little bit about the history of the early days of single molecules, going back nearly 30 years, and then how that became super resolution microscopy. I tell you about three-dimensional imaging, how we can look inside cells. It's really light and molecules. It's really electronic transitions of molecules. At its heart, quantum mechanics. So a lot of this does fit into a physics department, as well as the electromagnetic field work we're doing to give three-dimensional imaging. So this is changing the microscope to get new point spread functions for the microscope in the Fourier plane. And I'll tell you a little bit about that. I'll skip the three-dimensional version of single particle tracking and end up talking about tracking and trapping individual biomolecules in solution. So we have a lot of fun things to describe today. Let me just say that the historical part of this does extend back into 1987, 1988, 1989. And the real start was optically detecting single molecules at low temperatures. And this was done at IBM with a fancy optical technique, a spectroscopy technique called FM spectroscopy. But it arose out of the environment at the IBM lab, which I want to describe in just a few seconds. So the thing that you want to think about is, in those days, back in the mid-1980s, people thought that you could not detect a single molecule with light. It'd be too difficult. But we were inspired by the beautiful work that had been done in other laboratories to look at individual ions in electromagnetic traps. And so the question was, can you do it with a single molecule? Of course, there was a great physicist, Erwin Schrödinger, who said you cannot detect and study a single molecule, a single electron, or a single atom. And of course, that's a great lead, right? If a Nobel laureate says something cannot be done, then of course people should go out and try to prove the person wrong. So let me try to set the stage, though, to make sure everybody's on the same wavelength for us today. Yes, there's small jokes here and there if you're listening very carefully. Let's think of this molecule, pterylene, in a transparent host crystal of paraterphenyl. So pterylene has an extended conjugation, so that means it absorbs in the visible. Here's the optical absorption spectrum, excuse me, in the visible. And you see the first electronic transition here, the lowest energy or the longest wavelength transition. Now think about cooling it to low temperatures, because that's what a lot of scientists were doing in the 50s, 60s, and 70s. If you cool this to low temperatures, then what happens, 2 degrees Kelvin, these absorption bands get very, very narrow. Because we're no longer allowing any rotations or vibrations, and this particular transition is a zero phonon electronic transition. So the 0-0 transition has a lot of oscillator strength. I'm trying to say it in many different ways for many different contexts. That transition becomes extremely narrow, and you see 4 copies of it because they are in equivalent sites in the crystal. I've switched the direction here. Energy is going up on this one and to the left on this one. So anyway, this is the transition that was under great study. It's called an inhomogeneously broadened absorption. And at IBM Research, we were trying to use this absorption to store digital data in the frequency domain or the color domain. We were trying to write bits inside the shape of that line. Now, I won't tell you all the details about that technique called spectral hole burning. But the lead-in to single molecules during that time came from thinking about the fundamentals of optical storage at low temperatures. Trying to store data in bits and the color of the laser, writing different bits in the frequency domain. Now that's what the spectral hole burning idea allowed you to do for an inhomogeneously broadened line. But the point for today is that was the great flowering of these industrial research labs at that time. IBM, Bell Laboratories, General Motors, General Electric, various laboratories allowed you to do not only applications, but also the fundamentals underlying these ideas. And so it was one of these fundamental questions that led us in the direction that for today, asking and answering a question beyond the frontier of knowledge. So the question was this. I'm boiling it down to the simplest piece here. Think of one of those transitions I just showed you, one of these right here, and I'm showing it to you again. I've switched to pentacene, but that doesn't matter. It's another similar molecule. Here's that lowest electronic transition, and the question is, at liquid helium temperatures, is this a smooth spectrum? Is this a simple Gaussian-shaped smooth object or not? So that was something that we needed to know for the optical storage application. But no one had observed whether this was a perfectly smooth transition or not. And in fact, in 1987, we used FM spectroscopy to look at the shape of this absorption line in detail, and we saw this. We saw an amazing structure. And you might think, wow, this is noise. This guy's crazy. Tell me about noise. But it's not. If you measure it once and measure it again, you see exactly the same structure. This is what we call statistical fine structure. And it's coming directly from the individuality of the molecular absorptions, actually. Different numbers of molecules pile up at different frequencies. That's really what's going on, because each piece of sample is another measurement, another, if you like, roll of the dice to see how many are going to be present at each frequency interval. And so because this is coming from number fluctuations, its amplitude scales as the square root of the number of molecules in resonance. So that's a different spectral feature than most that you think about, right? Usually you put in 10 times more molecules, you get 10 times more absorption. But this effect scales as the square root of n. So if I put in 10 times more, it just grows by a factor of square root of 10, or 3. So that's interesting because, okay, it's an interesting physical effect. But in fact, when you think about it again, you say, well, wait a minute, since we can detect this, that means that going to the single molecule limit is only a square root of n more difficult, not n times more difficult. And that's good scaling, right? It's easier to get to the single molecule limit. So we pushed this technique called FM spectroscopy to the single molecule limit in '89. It's really very much like your FM radio, that you may remember. There used to be these things called FM radios, right? But it's being done here at 500 terahertz. We're using frequency modulated light, and we're using the single molecule to convert FM into AM. It's amazing. So the radio is the molecule. And so this is the signal. It looks a bit like a W because of a secondary modulation that's just for technical reasons. And so we at least got started with pentacene and paraterphenyl in 1989. But, at that time, people were thinking, well, or we thought, gee, this is neat. It's new, it's interesting, but how far could it go? We had no idea. You see, that's the beautiful thing about discovery and science. In a new regime, you never know how far it will go. A year later, after this experiment, Michel Horry in France also detected the absorption of a single molecule, again, pentacene and paraterphenyl, the same system that we showed, but he detected it by looking at the emitted light, the fluorescence emitted from the molecule. So that's actually gives you better signal to noise, so we use that technique now. So the beautiful thing about a new regime that I like to mention here is that a number of surprises immediately began to appear. And that's the kind of thing that you have really great fun with because you didn't predict them. One of them is that the single molecules would move around in frequency space, even at liquid helium temperatures, even in a crystal. That is, they spectrally diffused. And if you held the laser at a fixed frequency, then you see molecules jumping into and out of resonance. In other words, the molecule would turn on and off, blinking of its light. And that's going to be important later, but it was first observed here in 1991 in this experiment. We also could switch the molecules. We could drive them from one frequency to another at low temperatures. And that's partly by, if you like, stimulating the molecule, changing its local environment so it moves to another frequency. So the details of all these techniques were the meat of a lot of research in many labs all over the world, in the early 1990s. But very quickly, the field also then moved to room temperature because the idea was going to be much broader than just the low temperature measurements. And so in a number of labs, the single molecules were also detected at room temperature using the same idea of collecting the emitted fluorescence. I'll show you some setups for this a little bit in a few moments. But again, just to make sure that you learn about the surprises, we observed in 1997 that a single biological emitting object, the green fluorescent protein, something that is ubiquitous now in biological fluorescence, If you look at a single copy at room temperature of GFP, green fluorescent protein, you see it blinking on and off again, blinking on and off in some fascinating way. Now, I'm leaving out details because of time, but I want to just emphasize that the blinking really means that the molecule has a dark state accessible to it. So when you're pumping it and getting fluorescence, then it can sometimes go into a dark state. This could be either an isomerization, or could be through a triplet state, or could be other different mechanisms. But the idea is it will not emit, but then it can also come back and start emitting again. So that's what I mean by blinking. And seeing it for GFP was also a big surprise. So now, again, how do I summarize what went on with single molecules? And I can't. There's just too much that's happened. There's even single molecule experiments that are going on here and continue in many labs all over the world, huge numbers of applications of the idea. But what's the central element? If you watch one by one, then you don't have to average over millions of molecules or billions or gazillions of molecules. You can observe, are the individuals behaving differently from one another? And of course, that's very obvious when you have blinking and you have these processes that cause a molecule to turn on and off. If you have big assemblies, you would just see some slow decay. But when it's individual, it's very digital and so on. Gives you much more access to what's going on. But this is a short list of the number of things that have been done with single molecules. And maybe there's something in this list that you like or whatever. Optically detected magnetic resonance, surface enhanced Raman scattering, a lot of biophysics and cell biology applications. Why? Well, it's because in the biological regime, you already have very inhomogeneous systems. You have the potential that different enzymes are in different states, or that different motors are doing different things. And not averaging over many of them is a better way to see, in detail what's actually going on. But this is also true in polymers and other complicated systems. There's been work on quantum optics, photon anti-bunching, and that sort of thing. Of course, energy transfer. So just a huge list of single molecule applications. So to make sure now that we sort of collect and coalesce on one set of sort of optical experiments, to be clear, I want to remind everyone how the experiments are done at room temperature. We're again pumping a molecule from a ground state to an excited state, the first electronic excited state, but probably a vibrational sideband. There's quick relaxation down to the lowest electronic state, and then emission of photons that are shifted to long wavelengths. Okay? So we're typically collecting these long wavelength photons, and you want to cycle this many times to get many photons, right? So that's how you detect a molecule in principle. You tend to avoid dark states, which would produce blinking and so on, but you'll see an application of blinking in a few minutes. Just to, sometimes molecules are not so familiar to people. I mean, this is a wave function. The ground state has, obviously, electron wave functions. This is the first excited orbital, another wave function, and that's what we're promoting with an electric dipole transition. So the molecules that emit well are fluorescent-like molecules, like rhodamines or cyanine dyes or this green fluorescent protein that I mentioned. And the experiments at room temperature are typically done by attaching a fluorescent dye to the molecule of interest, and then you want to observe the light coming out of this label. Some biological molecules are automatically fluorescent, like the antenna proteins that are central to photosynthesis and some others. But in general, you can think of the experiments that work this way. Here's an example of how the optical setup works. Imagine focusing your laser down to a small spot. But you all remember that you cannot focus to an infinitely small spot. Due to diffraction, the minimum spot size is on the order of the wavelength divided by 2 times the numerical aperture. And numerical aperture is on the order of 1. Don't worry about it. Roughly, the wave nature of light prevents us from focusing to an arbitrarily small spot. So then how do you see these 1 nanometer sized objects? Well, you have to place them far enough apart so that you're not pumping more than 1 at a time. Now see, this was easy at low temperatures because we could use the color of the laser to select the different molecules. But at room temperature, we can't do that. So you must simply dilute more dramatically. And we'll solve this problem in a minute as well. Anyway, if you can do that, then the light that's emitted from that one can tell you that you've detected a single molecule and can give you information about what the molecule is doing. So a specific case here. Here's a case of looking at single molecules in a cell. And I want to dwell on this for a few moments because I want you to kind of appreciate what we do at room temperature now. If you do it this way, you see an amazing dance of the molecules. Okay? And these happen to be membrane proteins that have been fluorescently labeled with a dye, like I said. And it's called the MHC2 complex. That's part of our immune system. Very, very important in terms of the way we deal with invaders and so on. I'm showing you the upper surface of the cell here. So you see all these molecules are in focus. They're making a small spot. And the diffusion is being affected by everything about the membrane, okay? Cholesterol in the membrane, what's its concentration? We modulated cholesterol concentrations to change this diffusion and so on. You see molecules out of focus on the edge that are going down and out of focus. So you see that too. And then another thing you see from this old movie is that the molecules emit for a while, but then they turn off. What's that? Well, that turns out to be photobleaching, okay? Many of you will pay extra to get photobleached blue jeans, right? They used to be blue, but now they're white. You pay money to have the molecules be bleached. Here, it's a single molecule, so it's just on, and then bang, it turns off because a bond has been broken or it's been attacked by an oxygen or whatever. So that's another feature of room temperature that we deal with. You only get about 1 million photons before the molecule stops emitting. But nevertheless, if you use those photons in a smart way, then you can get a lot of information. So this regime is the one that's used a lot for single molecule experiments at room temperature. Okay, so now I've kind of given you several key elements that will now appear in a different context. By the way, you saw that these single molecules were not tiny dots of light. They had a size. That size is exactly that diffraction limit that I just told you about, a few hundred nanometers in size, okay? And so that's something that has limited optical microscopy since the beginning and leads to the Nobel Prize, which I want to talk a little bit about now. But before we talk about the Nobel Prize itself, I want to ask you a question. What is the next best thing to being picked for a Nobel Prize? Okay, so you might say, this guy's really crazy now. But I have an interesting answer to this question. My personal answer is being picked by Milhouse on The Simpsons. So this is true. If you go back to the 26th of September 2010, episode 1 of season 22 of The Simpsons, what's happening during that episode? Well, it turns out the family is waking up in the middle of the night. And they're going, oh, why do we have to get up so early? So they're like dragging into the living room, sitting on the couch, in front of the television set. And Homer Simpson says, because we got to watch the Nobel Prize announcements. And so why? Well, it's because they have a betting pool. They showed it on the TV. Here's the different family members. Here's the different prizes. And guess what? Yeah. So this is 4 years before the Nobel Prize. I was on The Simpsons, right? That's the greatest thing, right? Well, it also shows you that the Simpsons writers are not that bad. Here's Bengt Holmstrom. So he just won for economics last year. Okay. Here's Ben Feringa. He won in chemistry last year. So on this one slide, they picked three laureates. Okay, which is not bad, right? Well. Anyway, of course, it's more exciting to get the real Nobel Prize, but it's worth being on The Simpsons, I guess. So to set the stage a little bit for the impact of the Nobel business, remember we all have this grand challenge, and this is a grand challenge that's being pursued, even here with the beautiful imaging microscopy center that's just across the street. You want to observe the nanoscale machinery that's inside biological systems. And this happens to be a bacterium, which is already very small, a couple of microns long, maybe 500 nanometers across, a famous drawing by Goodsell. Looking more closely at a small piece of it, you see it's filled with these tiny structures. these little machines, these little motors. Part of it is cytoskeleton, part of it is DNA and RNA, and so on. And so there's basically a general challenge. You'd like to observe all of these objects and see what they're doing. Here's a list of imaging requirements you might think will be useful. I want high sensitivity and specificity in living cells if possible, and high spatial resolution. So spatial, you can imagine that you could meet a few of these things like spatial resolution with electron microscopy, which has very high resolution, but you don't have the specificity of fluorescence. You see, the things that I've been showing you with light and fluorescence and labels means that you've put the emitting molecule on the object that you want to see. That means the light is coming from the object only, the object that you want to see, okay? That's incredible specificity, and we have single molecule sensitivity. So what's wrong with light? What's wrong with light is that the smallest possible visible laser spot that you can make on this bacterial cell is too large compared to the objects of interest. You see, that's why some higher resolution is needed. So to say this a different way, or to kind of illustrate it a different way, this diffraction limit is, of course, the source of the problem. Here's my bacterium again. I'm showing you the fluorescence from a set of proteins that have been labeled with a fluorescent label. And you have to remember that you can't observe the structure just by buying the most expensive microscope possible, because if you did, here is what you would see from that, quote, most expensive microscope, and you don't see any structure. The image is fundamentally blurry because of that diffraction effect. Even though the emitters are just a few nanometers in size, they appear to be this few hundred nanometers in size. The diffraction limit recognized and written by Ernst Abbe in the late 1800s, lambda over 2 times Na. So what is super resolution? It is going beyond this optical diffraction limit, and you turn this image into that image. Now, I want to emphasize that this particular image is of a DNA binding protein. So you're seeing DNA locations here. But what's important to recognize is that this is 40 nanometer resolution or so. So it's 5 times beyond this diffraction limit. And I want to emphasize that this is not a small effect. We're not talking about 30% or 40% or 50% improvements. A factor of 5 is really huge in a fairly short period of time. And put it in the context, again, of electron microscopy. When the electron microscopists got to 30 nanometer resolution, that was super exciting, and many things could be seen. Nature and Science papers were flowing. And then they went from 30 to 20 nanometers, and another round of Nature and Science papers. And then they go from 20 to 12 nanometers, another round of Nature and Science papers. Each reasonable improvement caused much more to be seen. Here, it's just a factor of 5 in a short period of time. So that means that we have to be careful with these techniques. We have to think hard about, are we always showing the best possible image that we can? Are we thinking carefully about any systematic errors? And we and others have spent a lot of time thinking about those issues, of course, as it should be. So, of course, the prize it was to the 3 of us, and I'm happy to have shared the prize with these gentlemen. Stefan Hell surpassed the diffraction limit with a different technique than what I'm going to describe, because I'm going to talk about the way to do it with single molecules, because as you can see, with such a history in single molecules, that's what interests me the most, and that's also what Eric Betzig used. So how does it work? Okay? How do you do this? Well, first of all, you of course have to detect single molecules. We've shown that already a long time ago. Here's these images of individual single molecule emission spots, if you like. So the first key ingredient is what I like to think of as super localization. So what is that? Well, what it is really is simply spreading out this spot from a single molecule over multiple pixels of a detector. And you immediately see it has a shape. In one-dimensional cross-section, it looks like this. What this means is that the different pixels are recording different numbers of photons according to what I'm going to call the point spread function of the microscope, the fundamental response of the microscope. And it's in a standard microscope, it's an Airy function, but that's pretty close to a Gaussian. The point is, it has a shape, and we've sampled the shape by measuring the numbers of photons in these different pixels. So that means that you can take a fitting function and fit this data, the measurements, and constrain the fitting function. The width of it is still diffraction limited, right? We haven't changed that. But now you know the other parameter, not beyond the width, the center position, you know that much better. And the error in the determination of the center position has a much smaller distribution. The sigma of this distribution is roughly the Abbe limit, that big number, 200 nanometers, divided by the square root of the number of photons detected. Each photon landing on the camera, you see, is effectively a measurement of the position, and you did in measurement. So this improves as 1 over the square root of the number of measurements. So that means that if you collected 10 to the 4th photons in this image, you can localize the molecule to just a few nanometers in statistical precision. Okay, great. So that's wonderful, but remember this only works for an isolated molecule, one that's far from others, so that you can do this fitting business. And just as a note in passing, I just want to say that this process, this part, is an estimation problem. Statistical information theory is useful. We explore and understand this using those ideas. But note, I'm telling you about the statistical precision. I'm not telling you about accuracy. And so accuracy is extremely important, and that's one of those fundamental error analyses problems that we've spent a lot of time on and have dealt with pretty well in the past. But I won't go into detail about that. But it's worth noting that that's built in right here. Great, but it only works for isolated molecules. So what happens when you have a lot of molecules that are all labeling some structure? You need another idea, and that's, I like to call it key idea number 2, active control of the emitting concentration and sequential imaging. So here's the idea. We use molecules that have effectively 2 states. One state, well, I'm going to call it the off state. You shine light on the molecule, but it gives no photons back. The other state, you shine light on the molecule, and you do get photons back. So remember the blinking that I talked about earlier and the switching and so forth? That's where all this comes in. And here's how you use it. Here's the structure that's been decorated with fluorescent labels. That's what you want to observe. And if you just turn them all on at once, let them all emit at once, then the point spread functions from each of the individual molecules, or I'll call them fuzzballs, the fuzzballs all overlap and you don't see any structure. So you see how to solve it now. You just don't let them all emit at the same time. You force the concentration that's allowed to emit to be at a low level. So that, for example, suppose only 5 are allowed to emit on average. And you measure these 5, you localize them by this technique up here and record in the computer where they are. Let's say, and then photo bleach them or let them blink. The next time, a different subset will come on. Remember the stochasticity of the single molecule limit. A different subset will come on. And so you localize those and you do this again and again. And then finally, from the computer, you have all these positions written down. Show them all at once. You reconstruct the underlying detail. It's really a reconstruction. It's not an image, really. It's a reconstruction based on the sampling of the positions of the molecules at different times. Okay? So that's basically how this whole thing works. You might also note that you have to do this before the structure changes. This takes a little bit of time. It's like time domain multiplexing to try to measure the shape of the structure. So I first heard about this when Eric Betzig presented it as PALM in April 2006, photoactivated localization microscopy. Then STORM appears from Zhuang, FPALM from Hess, PAINT, DSTORM, GSDIM, BLINK, SOPHY. So all of these acronyms started appearing. And it was a flood of acronyms. Some people got confused by this. But in fact, they're all just different ways of achieving this on-off control that I was discussing. So it's the general idea underlying all this that really is important. Oh, we demonstrated it using the yellow fluorescent protein or green fluorescent protein, optical control that we observed in '97. We didn't put any acronym in that paper, so it wasn't too smart, right? If you leave out the acronym. So I'll give you one more acronym to put on the pile. It's a mechanism-independent acronym, Single Molecule Active Control Microscopy, or SMACM. Okay, so that's just for fun. Now, if there may be some who still don't quite understand how this all works, so let me give you an analogy that may help you explain this to your family or whoever you'd like to explain it to. Think about fireflies. Think about fireflies. So let me just turn that picture off and tell you, suppose you want to observe the branches of a tree and it's the nighttime, okay? You can't see the branches of the tree because it's dark. So you could just place fireflies along each one of the branches of the tree. Just place them everywhere. Never mind that they may not want to sit there or whatever. We'll ignore that fact. This is a thought experiment. But if there's many, many fireflies all over the branches of the tree, then you just watch what happens. They'll start turning on and off in a random pattern. You just take your cell phone, you take a video of these fireflies blinking on and off, and just remember that video, and then each time you see a firefly, you make a measurement of where it is in space, and then show them all at once, and you'll see the branches of the tree. Okay, so that's the way it works with fireflies. And now let's do it with molecules in a real cell. So on the left here, I'm showing you the standard diffraction limited image. And here's a cell, a mammalian cell. Here's the edge of the cell. Each of these spots is the light coming from single molecules. And these molecules are on an antibody that is labeling the tubulin molecules in the cell. So the tubulins have an antibody with antifluorophore, all right? And that's the light from those single molecules. On the right are the positions of all these molecules, right? You fit them and you place a dot where you found a molecule. If they're overlapping, then you just forget it. You throw it away for now because we'll keep sampling it again and again later. Next frame of the movie, I happen to have picked molecules that blink on and off, okay, quite dramatically because they go into dark states. This happens to be a molecule that interacts with a reductant that's added to the solution, a beta-mercaptoethanol. And so that reductant will cause the molecules to become, turn into anions and therefore not emit, but it's a reversible process. So the molecules will keep blinking on and off during the experiment. So there's the second frame, more molecules appearing. Third frame, more molecules. Next frame, more molecules appearing. And the whole video shows you the real story. Kind of like those fireflies I just mentioned. Here's the diffraction limited image, but over here is the super resolution image showing far more detail, okay, of the structure. So that's the idea. And so this super resolution now, over the last decade, has been applied to a large number of interesting questions in biology. Again, there's too many of them for me to summarize all of them. So I'm just going to give you a few examples, a very small number of selected examples. This is an axon, okay? In our neurons, the body of the neuron will send out an axon to go a long distance and make synapses with distant nerves. And so this is the axon where spectrin has been labeled. And you see an amazing banding pattern perpendicular to the long axis of the axons. This was discovered by workers in the Zhuang lab, and a structure that was observed by super resolution fluorescence and not observed before that. Here's an example of a nucleus of a cell where the DNA locations, the accessible DNA has been labeled by a labeling technique with resolution far beyond the diffraction limit. This is Huntington proteins. So there's a cell here. I'm not showing you the edges of the cell, but the disease, the brain wasting disease, amyloid disease called Huntington's disease, comes from mutant Huntington. And we can label mutant Huntington in model systems and observe what structures they make. And what was known basically before was that there were these large inclusion bodies inside these cells. But the super resolution imaging shows this huge array of fascinating tiny little fibrils that are all around inside the cell, all of them smaller than the diffraction limit. So we think that these ideas can have applications in the whole area of trying to understand these diseases that depend upon formations of aggregates in cells. And then finally, here's some more examples of these subdiffraction structures in bacteria, where here we've labeled the surface of the cell, and you can see these long flagella and stalks, objects that are smaller than the diffraction limit, so not easy to detect other ways. All right, so now think of super resolution as a technique, depending upon the schemes that I told you, but there's more to it. In fact, there's some fascinating chemistry involved because you need these molecules to switch on and off or be controlled by light. And so don't read the long laundry list, just follow me through these energy levels. What we want to be able to produce is molecules that are fluorescent, so that when you pump them, you get emission, but you want those in low concentration. That's what you need to get this super resolution to work. So one way to do that is to have a precursor molecule that's not fluorescent, but if you shine light on it, it converts into a fluorescent molecule. And you can do this arbitrarily weakly, so that'll work. That's called the photoactivation pathway. These other schemes that I've been talking about, or blinking and so on, involve going into some dark states like triplet states or anionic states produced by a reductant. So anytime you can have a set of dark states and can control the rates in and out, you can make most of the molecules be in a dark state and only a few in the bright state. So that makes blinking. But there's all these other mechanisms that are out there, including paint, binding-induced turn-on, chemical emitter generation, enzymatic generation, and so on. So there's lots of different ways to get this. Okay, so that means there's lots more to be done to come up with better molecules. But let's talk about optics instead. Let's talk about 3D, three-dimensional imaging. How do we do this? Well, we do it by a method called point spread function engineering. That's the way my lab has been focusing on this. And to explain this, let me just give you a reminder of what's in a conventional microscope and how we're going to change it. The conventional microscope has an object plane and an objective, and you collect the light from the object, send it through the microscope, and then there's a tube lens that produces the final image on the camera. In a conventional microscope, this region, which is called the pupil plane or the back focal plane, has nothing in it. It's just an aperture. And so that's what produces the point spread function response of this microscope. What I want to emphasize, though, is what happens when you move the object up and down in the z direction. If of course, you know that this is going to go out of focus very quickly. That's what you expect, right? But the point is, this response is not very good for determining the z position of the object. It changes slowly near the focal plane. It's a maximum near the focal plane. It's symmetric above and below. It blurs badly out of focus. So we've changed the microscope by putting something more useful in the back focal plane. What we do, one of the ways we do this is to put this phase pattern in the back focal plane. This is called the double helix point spread function or pupil function. And what is it really? Think of it just as a piece of glass with different thicknesses. And so what I'm showing you here in these colors is the radians of delay of the light that's going through different portions of the back focal plane. So it's really, you can make it out of a piece of glass with different thicknesses, or you can make this with a spatial light modulator that has a liquid crystal device in it that's changing the index of refraction, or you can do this with a deformable mirror. Any of those will work to produce a certain pattern, but what's special about this one is that it takes the light from a single emitter and produces two spots on the camera. Two spots. In fact, it's so interesting. It's an interference pattern. There's a null in the middle and there's two brights on each side. Even the light from a single molecule will do this, right? Because it's the electric field propagating before it gets detected. And so the other thing about this point spread function is that if I move the object in z now, watch what happens. The two spots revolve around one another. So if you think of this in 3 dimensions, it's like a double helix in 3 dimensions. That's why we call it the double helix point spread function. But the useful aspect is that if I have a molecule at a particular z, then the angle between these two spots tells me what the z is. I can calibrate in advance what different angles correspond to which z's. And so I get the x and y from the midpoint between the two spots and z from the angle. So that's how we get 3-dimensional images from this simple change to the microscope. And let me emphasize, we're not scanning anything when we do the 3D images I'm going to show you. We're not scanning. I just illustrated the different positions of molecules just for illustration. In one frame, we get all the molecules at the same time in a fairly thick piece of sample. In this case, this works and stays in focus over 2 to 3 microns in Z, which is a factor of maybe 5 or so longer Z range than you get with the conventional point spread function. So you see they have different angles between the two spots. All X, Y, and Z is all encoded in the shape of the spot. So it works. Here it is in bacteria. Surface of the bacteria is one color. A particular protein inside is the other color. And I'm just showing you that these are all full 3D images. Don't worry about all of the other stuff written here. It's just different, you know, even my students went crazy to come up with another acronym here called spray paint. But this idea of manipulating the light in the microscope has a lot of legs. A bunch of different groups, including ours, are inventing new point spread functions now that have different properties in terms of being able to modify the light coming from the microscope. And let me just emphasize that here's the conventional microscope that I showed you earlier. Here's where you normally put the camera, but you can implement all this with just another lens, the phase pattern, a second lens, and finally the camera. Very simple optics allow you to do this manipulation because it's being done in the Fourier plane in this 4F optical processing system. Here we're comparing a bunch of point spread functions based on what Z range they work over. Z range. Here's the double helix in the middle, okay, about 3 micron Z range. Astigmatism, which was an easier way in principle to get 3D, only works over very short range. And these new ones, saddle point, tetrapods, and so on, they can work even up to huge ranges, 20 microns in Z. Now, the price you pay for that is that the spots are much bigger. Okay, the footprint on the detector is larger when you want to have very, very long Z range. So I think these more intermediate ones are more useful. But I haven't really told you where these new point spread functions come from. This one called the tetrapod is of particular interest to us because one of my postdocs, Yoav Shechtman, calculated this point spread function based on an optimization algorithm. And so this now gets into one of the more mathematical parts of the talk. How do we compare point spread functions based on their fundamental properties and what is the most information that they will give us? We use ideas from statistical information theory, particularly a concept called the Fisher information. There's a function, or actually a matrix, a tensor called the Fisher matrix, and what it does is it expresses the sensitivity of the point spread function to changes in the parameters. It's a lot like the derivatives, second derivatives of the point spread function. And so what does this mean? Well, when the point spread function changes fast with x, y, and z, then you can do a much better job of determining x, y, and z. That's the idea. So the Kramer-Rao lower bound is essentially 1 over the Fisher information. So it's the minimum standard deviation you can get for determining x, y, and z. It's the best case precision. I'm plotting the CRLB on this axis, okay? And so remember, smaller values are better because I want smaller error. I want smaller localization precision as a function of z for x, y, and z, okay? Determining x, y, and z. And you see several different phase patterns here. Here's the double helix one again that I showed you earlier. Here's the phase pattern for the tetrapod. And these newer ones, the double helix and the tetrapod, are essentially flat. They're mostly independent of z. And in particular, the tetrapod was calculated to be the lowest CRLB that you can get from an optimization calculation. So anyway, that's some of this fun with point spread functions. Now, I now am at a difficult point because I'm either going to tell you a little bit more about a very special microscope called the tilted light sheet microscope, or I'm going to tell you about the Abel trap, this device that traps single molecules in solution. And so many people have asked me about the Abel trap today. I'm going to skip this light sheet thing, just showing you though, it's pretty cool. There's a tilted light sheet and there's all of these phase patterns I've just been talking about being used in an intrinsic way. These will produce 3D images in thick cells, including the lamin structure. So here's the lamin structure. This is the sac that's just inside the nuclear membrane, now rendered in 3-dimensional super resolution imaging over about 5 micron Z range. And it shows you that you can combine new ideas in the microscope as well as these new point spread functions to get fascinating, really amazing images. So going to this new thing called the Abel trap, let me just say there's another challenge out there. Beyond the imaging that I described to you, what if you want to watch a single molecule for a long time, but you want to do it in solution, where it's in a native environment for many biological systems. So the idea is we don't want to attach it to the surface or do anything. So we have this new device called the anti-Brownian electrokinetic trap. And here's basically how it works. You have a molecule that's in solution, and so it will want to diffuse away if you don't do something, Brownian motion. So what we do is use feedback forces to counter that diffusive motion. You essentially measure where the molecule is and then push it back with forces. So the forces we apply are applied, in different directions at each new time you look at it because it's moving randomly. So we have to have a closed loop feedback, electronics to do all this. The forces are applied by applying electric fields in 2 dimensions. So we put voltages on these electrodes far away, and they are what are producing the forces. In a microfluidic chip, so the whole thing is based on these voltages put in these wires. Here's where the action is in the center of this device. The z-dimensional motion is suppressed just because the trap is thin in the z direction, only 700 nanometers. And we're doing all this by having a laser beam that's moving around in the center part of the trap. So this was originally put together by Adam Cohen at the beginning just to demonstrate the idea, but Trinh Nguyen is the guy who over the last 10 years has enhanced and optimized the whole idea so that we can use it for looking at many, many different systems while they're in solution. So here's an example of what we can measure from the single objects. Remember, we're pumping and collecting fluorescence just like before. And so when the trap is on, you see a single object is just jiggling around the center of the trap. We can observe it for many seconds until it finally photobleaches, okay? But during that time, we can measure its brightness. That's a useful variable, and our measurement of brightness is good because we have a uniform time average illumination. So we can observe, let's say, one object that has 8 fluorophores on it as they photobleach one by one. We can measure the excited state lifetime by using pulsed excitation and looking at time delays. So that's the excited state lifetime for one fluorophore. We can measure the spectrum by dispersing the light. We can measure the polarization by collecting the light in 2 different detection polarizations. So 4 variables can be measured for the one molecule during the time that it's in the trap. That's the power of the method because this lets us use the idea for all kinds of interesting systems, mostly photosynthetic antenna proteins or redox enzymes or